Combinatorial chemistry: principles, methods, and applications. Combinatorial chemistry is a synthetic strategy designed to generate large collections of structurally diverse compounds, known as chemical libraries, through systematic and repetitive assembly of molecular building blocks. These libraries are subsequently screened to identify compounds with desirable biological or physicochemical properties. In contrast to traditional organic synthesis, which typically produces one compound at a time, combinatorial chemistry enables the simultaneous preparation of hundreds to millions of compounds, thereby significantly accelerating discovery processes in pharmaceuticals, catalysis, and materials science. The conceptual origins of combinatorial chemistry can be traced to the pioneering work of Robert Merrifield in the 1960s, who developed solid-phase peptide synthesis. In this approach, molecules are constructed stepwise on an insoluble polymer support, allowing sequential addition of building blocks and facile purification through simple washing steps. This innovation introduced the principles of automation and immobilization in synthesis and was recognized with the Nobel Prize in Chemistry in 1984. Building upon this foundation, modern combinatorial chemistry emerged in the late 1980s and early 1990s with the development of high-throughput methodologies such as multi-pin synthesis, the tea-bag technique, and split-and-pool strategies. Subsequent advances included the introduction of one-bead-one compound (OBOC) libraries, small molecule combinatorial libraries, biological display systems such as phage display, and DNA-encoded chemical libraries, which allow the construction and decoding of extremely large compound collections. At the core of combinatorial chemistry are several complementary synthetic methodologies. Solid-phase synthesis remains one of the most widely used approaches, in which reactants are covalently attached to polymeric supports such as cross-linked polystyrene beads and transformed through successive reaction cycles. The presence of a linker between the substrate and the support ensures stability during synthesis and controlled cleavage of the final product. Protecting groups, such as FMOC or BOC for amines, are essential for selective reactivity during multi-step synthesis. This method offers significant advantages in terms of simplified purification, compatibility with automation, and high throughput, although diffusion limitations and restricted reaction types can pose challenges. Parallel synthesis represents another key methodology in which multiple reactions are conducted independently but simultaneously, typically in multi-well plates using automated or semi-automated systems. Each reaction vessel yields a distinct and structurally defined compound, making this approach particularly valuable for structure-activity relationship studies and lead optimization in drug discovery. In contrast, split and pool, or mixed synthesis, is designed to maximize molecular diversity. In this method, resin-bound intermediates are repeatedly divided into groups, reacted with different building blocks, and recombined. This iterative process results in exponential growth of library size, often reaching millions of compounds. However, because the final products are obtained as mixtures, identification of active compounds requires decoding strategies. Solution-phase combinatorial synthesis provides an alternative to solid-phase approaches, with reactions carried out in homogeneous solution. This method allows broader reaction compatibility and the use of conventional analytical techniques such as NMR spectroscopy and chromatography during intermediate stages. However, it often requires extensive purification and can be less efficient for handling complex mixtures. A critical aspect of combinatorial chemistry is the identification of active compounds within large and often complex libraries. Addressable libraries produced by parallel synthesis allow direct identification of compounds based on their position. In contrast, non-addressable libraries generated by split and pool methods require encoding and decoding strategies to track synthetic history. Various encoding methods have been developed, including chemical tags, nucleic acid barcodes, and spectroscopic markers. DNA-encoded chemical libraries represent a major advancement, enabling the synthesis of vast numbers of compounds that can be identified through DNA sequencing techniques. The successful implementation of combinatorial chemistry relies heavily on specialized equipment and automation technologies. Robotic liquid handling systems, automated synthesizers, and multi-well plate formats enable high-throughput synthesis and screening. Analytical tools such as high-performance liquid chromatography, mass spectrometry, and nuclear magnetic resonance spectroscopy are essential for compound characterization. High-throughput screening platforms facilitate rapid biological evaluation, while computational tools support library design and data analysis. Despite the prevalence of automation, manual methods remain relevant in certain contexts, offering cost-effective alternatives for smaller-scale synthesis. Screening of combinatorial libraries can be performed using both experimental and computational approaches. Experimental methods include biochemical assays, binding assays, and cell-based assays, which provide direct evaluation of biological activity. High-throughput screening enables the testing of large numbers of compounds in parallel. Virtual screening methods, including molecular docking, pharmacophore modeling, and quantitative structure-activity relationship analysis, complement experimental approaches by predicting interactions between compounds and biological targets. However, computational predictions must ultimately be validated experimentally. Different combinatorial techniques exhibit distinct advantages and limitations. Parallel synthesis provides precise control over compound identity and is particularly suited for optimization studies, whereas split and pool methods enable the generation of extremely large and diverse libraries for initial lead discovery. Solid-phase synthesis simplifies purification and is highly compatible with automation, while solution-phase synthesis offers greater flexibility in reaction design but requires more extensive purification. The choice of methodology depends on the specific objectives of the study, such as whether the goal is to explore chemical diversity or to refine known lead structures. Combinatorial chemistry has had a profound impact on drug discovery and development. It enables the rapid identification of lead compounds from large chemical libraries and facilitates the generation of analogs for optimization. Early successes include the development of benzodiazepine libraries for central nervous system therapeutics. The approach is also integral to fragment-based drug design, where small molecular fragments are screened and subsequently combined to form high-affinity ligands. Beyond pharmaceuticals, combinatorial methods are increasingly applied in the discovery of functional materials and catalysts. Despite its advantages, combinatorial chemistry presents certain challenges. The efficiency of synthesis can be affected by factors such as molecular size, solubility, and functional group compatibility. The generation of racemic mixtures and the complexity of purification and identification in mixture-based libraries can also limit effectiveness. Nonetheless, ongoing technological advancements continue to address these limitations. Recent developments have significantly expanded the capabilities of combinatorial chemistry. DNA-encoded libraries now allow the generation and screening of billions of compounds with precise identification. Advances in biological display systems, including mRNA and phage display, incorporating unnatural amino acids, have increased chemical diversity and stability of peptide libraries. New synthetic methodologies, such as aqueous synthetic fermentation approaches, enable the construction of complex molecules under mild conditions. Post-translational modification strategies and macrocyclic peptide libraries have improved the development of drug-like molecules with enhanced stability and activity. Additionally, integration with computational chemistry has transformed library design, enabling virtual screening, machine learning-driven optimization, and incorporation of pharmacokinetic filters such as ADMET properties. In conclusion, combinatorial chemistry represents a transformative approach in modern chemical science, enabling rapid exploration of chemical space through high-throughput synthesis and screening. Its continued evolution, driven by advances in automation, encoding technologies, and computational methods, ensures its central role in drug discovery, chemical biology, and materials innovation.